All right, so the third part of our discussion about translation. The first part we talked about the genetic code and understanding some of the features of that code, the codons and how they're read. Second lecture we talked about the basic process, but in looking at the units that were involved, tRNA, tRNA charging, what a ribosome was from a structural standpoint, and of course we've already talked about message, the other third part. Finally, we're going to actually do the translation. Now this is going to be cartooned as we've done for almost everything else. Remember, this is an introductory level course, so we're just trying to go through the process of expression and cell biology, biochemistry, other things will fill in all the fine details about what's going on. Molecular genetics, if you're fortunate enough to take that later on, will fill in all these parts. So we're trying to just give the general model, and it is sort of a hybrid model. So it's kind of mixed in of various ways that it happens in different organisms. This is sort of the, if you would, this is sort of the syndrome part. This is the collection of the way it kind of looks most of the time. Not everything will do it this way exactly, and many other things will have many other additional components, but it gets the point across. Okay, so we did activation. We talked about this charging of tRNAs and the specific charging, and then some of the things that can actually go wrong. So now we want to actually get into translation. Translation will have three parts. It will have an initiation phase, elongation phase, and a termination phase. Initiation is really when we start building the ribosome, as well as connecting that ribosomal structure to the message itself and bringing in the first elements of the charged tRNAs that we're going to use. So let's go over the little schematic. So here's a piece of messenger RNA. It's been matured. It's got a 5' cap alpha here. It's got a 3' poly A tail off here. Now, I want to point out, and you'll see this as we go through this, AUG is the start code, but there will be many bases ahead of that AUG. There will be a non-translated region that is out in this direction, okay? So the cap's off out here somewhere, and there'll be a number of nucleotides that depend on the message, but eventually there'll be an AUG. Now what's important about that is that the initiation involves the small subunit coming in and sort of moving along and actually finding that AUG. So in many ways, the actual code that's read is framed by this small subunit finding this AUG. If it misframes, if it finds it UGC and it still puts, that becomes the first three letters. Remember, it's a blind process. Once you define the first three letters, it is just sequential. So the next three letters become the next codon, the next three become the next, etc. So it's critical that this unit here, this part of the small unit of the ribosome, that it locate this properly. And it does that by some of this RNA. Remember, there's an RNA strand that's actually built into all these proteins that make up the small subunit. All right, but in initiation, the first real thing that happens is that you have an attraction of this small subunit. Now that takes a lot of other proteins to work. Remember when we went back to replication and we go back to translation or transcription, there were other proteins that came into place first and attracted the system. Well, that happens here. We'll look at those more in a moment, but I'm just trying to go through the basic process, okay? All right, so once initiation occurs, then you have out here, you'll have the formation, partial formation of the large ribosome. But the way that it's normally presented is that the next real thing that happens is that the first tRNA comes in with the anticodon for AUG. And of course, it will be carrying methionine because that's the first one. And it'll come in and fall into this P site. That's the only time that a tRNA comes into the P site directly, okay? That's the only time a tRNA comes in directly. From now on, they'll be coming into the A site over here. All right, as part of that process of bringing that in, you get the formation of this whole large complex that makes up the full ribosome. So now you've got the 40 Swedberg unit here, the small subunit, the 60 will come in now, and you'll form a complete ribosome. And that complete ribosome, as part of this process, will have this tRNA associated with it bound into the first position. Okay, now we're ready to start moving. That's initiation. Initiation has occurred, and we can go backwards here. Initiation has occurred when you have small subunit come into place. Again, there's a complex of proteins that are involved to make this work. You have the first tRNA coming into the P site, and that's the only time you'll come into the P site directly. Okay. That all happens as far as catalyzing the final formation of the full ribosome by bringing in the large subunit. And at that point, you now have the initiation process. You're ready to go. Okay, so from now on, what you're going to do is you're just going to start bringing in tRNAs to the A site, and you're going to ratchet this message along through the process. Okay, so you're going to ratchet this message out. You're essentially going to, the ribosome is, if you will think of it as moving, but this message will come, and the next codon will be in place, it'll be filled, and then we'll move to the next codon, the next codon, the next codon as we do elongation. Your book shows it something like this. Okay, obviously T charging has occurred out here, and all the amino acids have been attached to their appropriate tRNAs. The small subunit comes into play. You bring in the, again, you see this nice region here, which is untranslated. You find the start code. You bring in the first appropriate tRNA into the P site shown here, and then that catalyzes the attraction to bring in this whole unit. Right now, something really important happens here. As I said, it's lots of stuff going on. Your book shows it much like this. Okay, your book. Remember that poly A tail? I told you the poly A tail had several jobs, right? Part of it was to protect the end of the message. So the longer the poly A tail, the longer the message will persist or be used. Other things that can happen, and of course, it helps signal that you should move it out of the nuclear region into the cytoplasm. And its third job is to actually fold back and help cap this ribosome itself. Okay. Now, unfortunately, your book draws it in a way that's not completely accurate by what we currently understand. We currently understand that the small subunit has already found this AUG, and that the ribosome then builds, and these capping proteins, which are held on by the poly A tail, are complex into that. They're there to help the small subunit actually move along and find this, and then once they're there. So in reality, it looks more something like this. Remember that cap that's out here? There's specialized proteins that are associated with it. Your 40S unit is attached down here, and this is starting to cap up, and all this in here is the building of those of that larger subunit of the ribosome. So the tRNA would be, this is the untranslated, this is your start code. So it's going to actually move along down through here until it finds the first start codon. tRNA is going to come in, and then that'll complete this complexing up here. So while your book draws it like this, that's really not what happens. This implies the ribosome forms completely and then moves down. That's not, it's all sort of a synthetic activity that occurs as a continuum. All right, just wanted you to understand that, so that if you go into cell biology or to biochemistry and you go back and go, well, this is what he told us and this is what our book said. Well, it's correct in this partial sense that all happens, but it happens in a different pattern. All right. All right, well, now for us, we've got it initiated. We've got the large and small subunits bound up. We've got the first P site filled in. Now we're just going to bring in these charged tRNAs. We're going to use GTP as our energy source, and we're going to start to fill in all the appropriate amino acids by bringing in the tRNAs into the A site, the acceptor site, or the amino seal site. Okay. We're going to have an enzyme known as Peptidyl Transferase. Once again, this is a complex of proteins that's part of this large ribosome. It will catalyze a bond between the amino acids known as a peptide bond, right? Because we're growing a peptide. And at the same time, it will help catalyze the breaking of this amino seal bond that was over here to this tRNA in the P site. So it will do something like this, okay? All right, once it does that, what happens is the entire ribosome kind of ratchets down through the message. So we will now move this A site down to this next 3 letters. Remember, it's just a blind, commaless process. And we will actually leave this in place, but it'll move over here. The P site will now be occupied by what was in the A site. This will move to what's known as the acceptor, or as the ejector site. Okay, I've sort of drawn it off here out of the way, but really what's going to happen is this is going to stay in place, but it's going to be moved over to a third site. Okay, and the ribosome will move like this, and that discharged tRNA will still be here. It'll just be in the ejector site and thrown out. Okay, so, all right. So now we've got the P site filled. the A site is available. And so elongation is nothing more than reading this code, bringing in the next tRNA. Aminoacyl synthetase will catalyze a peptide bond, and you'll now have a growing, you have a tripeptide here. It will catalyze the breaking of this bond over in the P site right here, and you'll get a longer structure, okay? And we will move and we'll start to ratchet again and ratchet again. So this keeps moving down, okay? This is probably a better view of what's going on right here. We have the A site, P site, and this, again, this ejector site looks something like this. The growing polypeptide actually grows out of the acceptor site, out of a tunnel, if you want to think of it that way, an area that can, it's where it can just be moved through the process, the A site. And then you ratchet over, and once you ratchet over and this charged tRNA ends up in the ejector site, then it is literally ejected. It is pushed out of this, ready, available to be taken up and recharged in some form. Okay. Again, all this activity that's going on here, this movement activity and stuff, is being driven primarily by GTP, the phosphate groups that are carried there, as opposed to ATP, which is another, the very common energy carrier for our most cell systems. All right, so there's our site. So we're growing this along. We're still in elongation. We come along, we find the next one, and if it's, however many codons you have here, however long the gene was, it might be 60 or 70 amino acids that are added. It may be several hundreds of amino acids that are added, but you just keep adding them through the same process, forming these peptide bonds, releasing these amino-seal bonds, and ratcheting over. Okay, at some point, the A site is going to be occupied by a term code, one of those 3 term codes, UGA being this one, UGA, right, UGG, the ones that you've learned, right, from the coding dictionary. We said term code, there are no tRNAs for that. There's no anticodon tRNA for that, so it's an empty site. So what happens here is we initiate the process of termination. The A site is part of this recognition is that there is a stop codon there, okay? And what happens is that this signals the bringing in of release factors. Okay, factors again are just small proteins that come into play. They occupy the A site, and as opposed to a tRNA, they cause a release of this tRNA that's been connected here. That's part of the process, and that's part of catalytic breakdown of this bond, this last bond that you had here between the very last amino acid and the very last tRNA. That frees up the polypeptide, also frees up the tRNA, and it causes, with some energy, dissociation of these 2 subunits. Okay? So they're finally released, and their ribosomes are now free to go back and form up again and to be reused as a factory. Okay? And so this is termination. You've got these release factors. You have to break this final bond right here, as well as release both the polypeptide itself and the tRNA. Okay? Your book demonstrates, the whole process something like this. We have charging, small subunit, large subunit. That's bringing in the first tRNA with MET. Is end of initiation. Once you have initiation, you simply move down, making larger and larger groups of tRNA, bringing more and more in, making larger numbers of connected amino acids to make your growing polypeptide. And eventually you hit a term code and you stop. All right. Now again, the poly A tails back up here, it's being modified. All these things are happening. Now in some messages, we've actually already started to do a translation again. Some messages are used multiple times, and you can have simultaneous translation, so that on this end down here, once it's free, you get recombining of this. So you can actually start the whole process over again. So others wait till the end, and sometimes messages are only used one time and then they're degraded. It just depends on the message. But there's multiple things that can go there. All right, so again, I realize this is cartooning it, but it gives you the major steps, right? It gives you the tRNAs, how they come in, and how they make these polypeptides that are critical to us, because it's those polypeptides that we're going to make eventually into proteins, and it's those polypeptides that give us phenotype, right? That's where we're trying to go in genetics, right? The next thing we'll do is maybe just talk a little bit about some little problems that you might try to work and understand these. How do you study this stuff? Go back and learn the 4 major steps. Go back if we were to show you this picture right here with some of these things blocked out. We'd say, where's this? You'd say, it's in elongation. Oh, by the way, it's happening during translation, not transcription. And what is this thing here? Well, it's the ribosome, or what's this? So, you could just take any one of these pictures, take away the labels, and put your own labels back in it. That would be a good way to study the process itself. And of course, it's like a movie, right? I've told you a number of times with replication, transcription, translation, they are a movie. So kind of in your mind, be able to stop them at a frame, say, okay, what's going on in that frame? What are all the pieces that should be there at this point? What just happened, what just left, and what's just about to happen? And what are the pieces, the next things that come in? Once you can do that, you've got these really well prepared for pretty much anything we could ask you.